Hello students, I am going to explain now about the Avery et al. Experiments. In the year 1944, Oswald Avery and his colleagues Colin MacLeod and Maclean McCarthy performed an experiment in the search of the substance that was named as the transforming principle by the person whose name was Griffith. Griffith was not able to pinpoint what was the substance that had changed the R2, leaving R2 into leaving S3 by taking the substance that is known as transforming principle from the dead S3 cells. So let's continue with the steps. As you can see, he along with his two colleagues performed the first step, in which three of the Griffith's experiments steps used to kill the S3 capsular smooth stain by boiling its suspension to heat kill it. This has formed the S3 cell lysate. Lysate is nothing but dead cell suspension, which is crucial for the next steps of the Avery et al. Experiments. So as you can see, Avery et al. Performed the third important step in the Griffith experiment, that was to kill the S3 by heat and forming its lysate. As you can see, by using the S3 cell lysate, which was heat killed S cells, Avery et al. Performed the following steps. In the first step, they took some S3 lysate suspension, added to it proteases to destroy the protein in the S3 lysate, plus they have added the living R2 cells suspension into the killed S3 lysate treated with protease. So what happened after mixing the living R2 with protein destroyed S3 lysate? The living S3 cell colony still appeared, means something was there that has converted or transformed R2, that is taken by R2 from the S3 lysate. In the second step, Avery et al., means Mr. Avery and his colleagues, took same S3 lysate from the initial step and now added to it carbases. So carbases destroy carbohydrates. What happened now after addition of carbases to S3 lysate tube suspension? The all carbohydrates that were present in the suspension were destroyed. The living R2 cells were added to this carbase treated S3 lysate and mixed to allow the transformation. What happened? Again R2 converted and living S3 cell colonies appeared on the bacterial culture media. So this was also surprising. After protein destruction also, the R2 converted into S3. After carbohydrate destruction also, the living R2 converted into living S3. Then in the third step, the S3 cell lysate plus lipase, means S3 cell lysate is treated with lipase enzyme which destroy the lipids in the S3 cell lysate. Now it is mixed with living R2 cell, which thing? The S3 lysate treated with lipase is now mixed with living R2 cells. Again after transformation, surprisingly, living S3 cell colonies appeared onto the bacterial agar. So they performed two crucial next steps. Let's see. As you can see, in the third step, the Avery et al. Performed destruction of lipids from the S cell lysate, but there was still transformation occurred of R2 cells to living S3 cells. So they have performed the fourth step here. S3 cell lysate is now treated with DNase enzyme which destroy particularly DNA. When the DNA is destroyed, the S3 cell lysate is mixed with living R2 cell suspension and allowed to transform. The living R2 cell colonies only appeared after the transformation. Means actually no transformation occurred because there was no living S3 colonies. So from dead S3 cell, when it is treated with DNase, there was nothing available to transform for the living R2 cells, as you can see in the diagram. So this was the main step of the Avery et al. Experiment. Even then they performed the next fifth step, in which the S3 cell lysate is treated with RNase enzyme which destroyed RNA of the S cell suspension of heat killed S cells. So even after treating with RNase, the living R2 cells were mixed and the living S3 colonies again appeared. So as you can see on the board, when protein is destroyed, there was transformation. When carbohydrate is destroyed individually, there was transformation from R2 to S3. When lipid is destroyed, there was transformation from R2 to S3. When RNA is destroyed, there was transformation from R2 to S3. But when DNA is destroyed, there was no transformation of living R2 into S3 cells. Means the DNA was the substance known as transforming principle. If DNA is there, then only the transformation can occur of living R2 cell into living S3 cell. Here in the fourth step, the DNA is destroyed by DNase enzyme. So what happened? Due to destruction of DNA, the transformation has not occurred. So again they have added the purified DNA of S3 lysate and mixed it with R2 cells. The R2 cells converted or transformed into the S3 cells. Means it is confirmed that the DNA was the transforming principle discovered by Griffith in the year 1928. Bye bye. I will come in the next video with next point.